Good morning, everyone. Thanks, Sarah. I feel great. I hope you all do, too. And it's great to be back here. I really appreciate the invitation to come back to Duke, where I spent 15 wonderful years, and the Lemur Center was really a big part of that. And I hope you see today, although my work is broadly covers many different areas, that the Lemur Center has figured, I think, prominently in the way that I approach my work. Now, I think it goes without saying, we all appreciate the fact that we and other primates are highly visual creatures, right? So that is, in fact, one of the defining features of primates is a high reliance on vision. And for us, we use our eyes to direct our fovea, the high acuity portion of the retina, at objects of interest in the visual scene. So if you're looking at this scene, for example, you might be looking at the high, the brightly colored things, the high contrast things, and in particular, the people who are in the photograph. And this intense interest in paying attention to others is really important for us. Not only are we motivated to direct our attention to other individuals, but we use their attention. This is something Brian talked about yesterday, gaze following. We use the attention of others as an informative tool that we can use to gather information that might be relevant for us. So we tend to, when we see somebody looking at something, we tend to look at the same thing. Attention to others is critical for the development of normal human cognition. So typically over the first year or two of life, infants begin to look at other individuals, principally their caregivers, and then begin to develop gaze following and joint attention. And this is really critical. Sharing attention with others is really critical for developing an understanding of the minds of other individuals, okay, for developing a theory of mind and subsequently for developing language as well. And we now know that the failure to develop normal social attention has profound consequences for disorders like autism, in which this typical interest in others and typical tendency to share their attention begins to fall apart during the first year of life and seems to have subsequent consequences on other aspects of behavior, in particular understanding the minds of others. Oops, that went a little bit farther ahead. Now, in order to really understand social attention, how we use visual information to guide our attention to others, we have to actually understand the behavior. And it turns out that that's pretty tricky to do, to actually measure orienting behavior. Of course, the eyes, when they move, saccadic eye movements are very fast and ballistic. And to really know with any precision what an individual is looking at, I think, is very challenging. And in fact, the scientific study of visual orienting behavior did not really take off until the middle of the 20th century with the work of Alfred Yarbus, who's a Russian psychologist. And he developed a very ingenious method for measuring precisely what people were looking at. It demanded serious heroism from his participants. He basically he took tiny little mirrors and he suction cupped them to the eyeballs to the sclera of the participants, and then he had them look at pictures, photographs, paintings, or objects like this one, and then the reflected light would be traced out on photo paper. And you see here sort of typical gaze scanning pattern of an individual looking at the face of a girl, and you can see the typical pattern of attention to the eyes and mouth. So Yarbus did many, many studies, published a couple of wonderful books, and I really like this example here, which shows the sort of intrinsic value of social information for guiding attention and how that can be modified or traded off depending on the goals of the individual. And so this is a trace from an individual who's looking at a painting called The Unexpected Guest, and on the left is the trace of attention of eye movements when the participant was just asked to look at the painting, and you can see that the participant is basically looking at the faces of the people in the painting. But on the right, the participant was asked to guess or to estimate the wealth of the family, and you can see that the pattern of eye movements changes quite a bit to examine the clothing basically worn by the individuals. And so this, I think, illustrates the principle that, in general, we are intrinsically motivated, intrinsically interested in acquiring information about others, but that can be modified depending on our goals. Okay. Of course, now we have much better, easier on the participant techniques for measuring orienting behavior. So in the last 10 years, there's been incredible development of technology that relies on infrared video, high-speed infrared video that can, at a distance, actually measure your eye movements very precisely within arc minutes of rotation and up to 2,000 times a second. And you can literally buy these things for just a few $100s, and you can even buy wearable versions of this that will allow you to move about the world for under $2,000. So it's been quite a remarkable change in technology. And with this kind of technology, scientists have been able to go further into understanding, say, the differences between typically developing individuals. So this is a lovely study done by Ami Klin about 10 years ago. A typically developing individual watching one of my favorite movies, Who's Afraid of Virginia Woolf. And these two are talking about him, and so you can see that not only does the participant look back and forth between their gaze, but they are also interested in his reactions. And an individual with autism, for example here, whose scan path is traced out in red, does not show any interest in those typical features, okay? So if we are going to really come to some understanding then of the mechanisms that mediate our own visual orienting behavior, particularly in social situations, of course, we are going to need to actually understand what's going on in the brain. Eye movements, visual orienting behavior is not generated by the retina and the eyes, but actually passes through the brain, okay? So it all happens here. There's 3.5 pounds of meat between your ears, and of course the brain is made up of about 80 billion neurons, okay, making maybe 100 trillion connections with each other. And so when scientists really need to understand, when we want to understand the mechanisms, the real biological mechanisms that mediate this kind of behavior or any other kinds of behavior, we have to get down to this level of analysis, particularly if we want to develop new therapies that might improve impairments in visual orienting behavior as attend autism. And the standard approaches for doing so in an animal model really kind of fail to apply here. So despite all the wonderful molecular genetic trickery that you can apply in a standard model like a mouse, really doesn't work here because mice are not particularly visual at all. They don't make saccadic eye movements, and their social lives, I would submit, are not particularly interesting as well. And so that means that we are going to, learn a lot more about ourselves by studying both the behavior and the biology of other critters with whom we share these features, and that means non-human primates. And what I'm going to show you here is just a video. Oh, you have the video player. So if you'll press play, this is just video of a rhesus macaque who's sitting in the laboratory. Actually, it's video of several rhesus macaques watching an ethologically relevant video of monkeys who live on an island off the coast of Puerto Rico, island of Cayo Santiago. And basically, we gave monkeys, I hope this is going to play. Okay, it's not playing. Well, what you would have seen had the video worked, and I hope the other videos work. So each of those colored dots is the point of regard. It's where a monkey participant's gaze is pointing at any one time. Where there are numerous dots of the same color, that indicates that the same monkey had watched the video many times. And the different colored dots are for different monkeys, okay? And what you'll find is when you have monkeys watch videos like this, they're basically like people. So they tend to look at the faces of other monkeys. They're really interested in monkeys eating things and drinking things, and they also spend a lot of time looking at the rear ends of other monkeys. And that makes a lot of sense. If you know anything about rhesus macaques, like many non-human primates, they convey a lot of information about the reproductive state through their hindquarters, okay? So there's signaling information there. Okay, and so in a number of studies, we have set out to try to actually quantify the value of social information for people and for monkeys, and I would submit that both of these two stimuli here convey a lot of information. So the one on the left, this is obviously a human being, a young woman, probably of high reproductive potential. And for those of you who know something about rhesus macaques, the same can be said for the one on the right, which is a young female of high reproductive potential. And we would postulate that actually this information has value, and if it has value, then individuals will pay for it. And so this is, we actually tested that idea in a study we termed our pay-per-view study. We basically asked monkeys to make choices between two yellow boxes on a screen, and if they chose one of the boxes, they would get a squirt of fruit juice. If they chose the other box, they'd also get a squirt of fruit juice, but they'd see a picture, and those pictures could be drawn from a huge array of photos of the faces of dominant males, subordinate males, female hindquarters, etc. And then we play a game where we would either pay them more or less to choose the picture side. Then you can plot the proportion of choosing the photograph as a function of its relative payoff in juice. And monkeys are smart little economists, and of course they're going to choose the one that pays off more often than the one that pays off less. But the curves will separate, as they do here in red and green, when the images actually alter the value of the particular option, of that option. Oh, that's... Okay, I could try to give this talk. This is a game that we used to play in the Center for Cognitive Neuroscience, where we'd get a random slide deck and try to make our way through it, sort of science karaoke here. Okay, we're back. We're live. Okay, so the red curve is when the images were female hindquarters. Per any of these are males who are making these choices. You can see the curve slides to the left. If you read off the PSE, the point of subjective equivalence, that's how much juice that monkey will give up to see those images. The curve in green is for pictures of subordinates. It slides to the right. You have to pay the monkey extra. And that's what you see basically on the right, data from a bunch of monkeys viewing these different classes. You can see that basically information about sex and status has value. Monkeys will give up juice to see it, and you have to pay them to see subordinates. And actually it turns out that Duke University undergraduates are also motivated by visual information about others. In this case, we used images of thousands of males and thousands of females that had been downloaded from the website hotornot.com and evaluated for physical attractiveness by a separate set of raters. And then we played the same game, but the choosers were choosing based on money. So they're winning or losing different amounts of points that could be traded for money at the end of the game. If they ignored the faces, they could take home an extra $7 about on average. But they tended not to ignore them, especially our male subjects, male participants, and they would typically pay on average about 1/4 cent for each opportunity to see a female who was anywhere from average to high physical attractiveness rating. Okay. The important point here is that here both in monkeys and in people, there's intrinsic value to social information. We can quantify it and we can use that as a tool for probing its representation in the brain. Now, it turns out rhesus macaques also follow the gaze of others, as you heard from Brian yesterday, and now it turns out that many animals follow gaze. This is from a study in which we compared the gaze-following behavior of humans and rhesus macaques. Basically, they just had to shift their gaze to a target as quickly as possible, but then we would flash up a face of a rhesus monkey either looking left or right. And what you're seeing here comparing green to red are when the face of that monkey was looking in the direction the target would pop up. Both people and monkeys tended to be faster when their expectations were met. That is, when they had shifted their attention in the same direction as the monkey. And we also saw tiny little microsaccades going in the same direction, which are consistent with the idea that just seeing an individual's face or eyes pointing in one direction or another is enough to provoke a process that shifts attention in that direction. Now, we now know from a decade of work, at least, using non-invasive brain imaging in humans, that this process unfolds in this fashion. So after visual information comes in and is processed in primary visual cortex, it gets relayed to the temporal lobe, which is activated by the sight of other individuals, basically seeing faces, seeing bodies, seeing moving eyes, things like that. That information is then relayed to parts of the brain in the front, okay, like the orbital frontal cortex and the ventromedial prefrontal cortex, in which my colleagues and I here, Scott Hutel in particular, showed that the value of this social information is represented in those areas, and then finally that information is relayed to parts of the brain involved in shifting attention, primarily the parietal cortex. For example, when you see a face turn and look in one direction and your attention shifts, that seems to be mediated by the parietal cortex. And it turns out from our work and the work of many other individuals that in the temporal cortex of monkeys, there are individual neurons that are specialized for detecting and decoding faces, their direction of gaze, and also the hindquarters of other individuals. There are motivational signals that indicate the value of that information in these same parts of the brain that have been identified in humans. And then anytime a monkey's attention is motivated to be shifted toward a social target, we see engagement of neurons in the parietal cortex. Okay, I know that's been a 10-minute detour. around to lemurs, but now is where the interesting things come in. So this raises a lot of interesting questions. So here we have shared, I would argue, homologous circuits in the brains of ourselves and rhesus macaques that motivate this social attention, this attentional process that we develop early on in life. And we know that we shared, last shared a common ancestor with rhesus macaques some 40-some million years ago, 38, 40 million years ago. But it raises, I think, the really provocative question as to whether these circuits might be even more ancient or whether there might be convergence, basically latent circuits that belong to, that developed much earlier, evolved much earlier within the primate clade. And this is where lemurs come in, where we can ask the question whether these animals also seem to be specialized for the same kinds of visual orienting behavior. And that's where the Lemur Center comes in. I had this question about, I don't know, maybe it was about 12, 13 years ago, and maybe it was longer ago than that. And I thought, well, we could ask that question out here at the Lemur Center. It would be really cool if we could really measure eye movements in lemurs with high precision using the technologies that were being developed at that time. And Bill Highlander was the director at that time, and he said, sure, we can do that. And our animal care staff would be really happy to help you develop these techniques. And so we focused on ringtail lemurs because they're the most gregarious, the most social of all of the lemurs here at the Lemur Center and in general. And we applied a technology that was at that time in its infancy, which is using telemetric infrared video-based eye tracking. And this is the state of the art now. At that time, that was not the state of the art. In fact, these eye trackers were just being developed, and we worked with a company called iScan to develop. This is the first measurements of visual orienting behavior in freely moving animals done anywhere in the world. Okay, this was done here at the Duke Lemur Center. And what we did is we trained two lemurs, Licinius and Arrakis, to first wear a little vest, and then they would gradually learn that there would be something in the back of that vest, which was the video transmitter with a big heavy battery. And then we had a very lightweight set of cameras that we would basically Velcro to their heads. They had a little helmet, a little bicycle helmet that they would wear. And one camera pointed out at the world, the other camera bounced some infrared light off of a see-through mirror and recorded the position of the eyes. Okay, so you can imagine that this was pretty challenging to do. We had to be very careful about the state of the animals. The animal care staff here were really wonderful in helping us to develop this. And then we just let them out into these natural habitat enclosures or some of the larger arenas. And what was amazing is that this wonderful thing about lemurs, they just they forgot that they had this equipment on, and so they would, these guys actually got in a big fight here. Nothing bad happened, but they were running back and forth and doing their stink fighting, etc. And what was really cool is we could measure what the lemur was looking at during those kinds of encounters. And I'm really hoping this video is going to work, because otherwise my thunder will be stolen. Can you try to play the video? Please. Okay, good. So this is just a very short video clip. The red dot is where the lemur is looking. You can see there are two individuals who are having an interaction over there through the chain link fence there, and the lemur, who was basically stationary, was spending most of his time looking at them. Okay, maybe no surprise, but we're doing this very accurately. And what we found is that basically measuring orienting behavior across really many, many hours of these kinds of clips. And I will also point out that this still poses a huge analytical problem. Really, we are on the cusp of being able to do this in an automated fashion using computer vision techniques, but basically undergraduates had to, frame by frame, indicate where gaze was and where everything in the scene was. And this really crushed the careers of budding young scientists here at Duke. So I feel really bad about that. But what we found is something that's very similar to what we found, we and others have found in people and in monkeys, which is that here in blue, when the lemur participants, when Arrakis and Licinius were stationary, they tended to spend most of their time looking at other lemurs, okay? It's pretty interesting, a lot more time than they spent looking at food, for example. But when they were moving, then they stopped looking at other lemurs and they tended to look along the routes that they were about to traverse, especially when they were arboreal. This is, I think, really interesting. It shows that their visual orienting behavior, just like our visual orienting behavior, is very sensitive to our goals and the demands on our behavior at that time. This is some work that we're following up on now with Dan Schmidt. Okay, how about gaze following? I also said that this is a really important, right, behavior for humans and for rhesus macaques. Brian told you a little bit about it yesterday. It looks like these guys are looking at the same thing, right? How about during ongoing behavior? Can you play this? This is a little bit harder clip to kind of piece out, but gaze is in red, so he's looking at the animal care staff here and then turns and looks right there to the area in which the other lemur turned his head. And if we actually trace that out, we see that it's very important to do this, to look at the temporal sequence of those movements. And it turns out here in blue is when the observed lemur turns and shifts his gaze, and we see that, I don't know, about a couple hundred milliseconds after that head turn, then the participant lemur shifts his gaze in the same direction. So looking very much like gaze-following behavior. And in fact, when we looked at all the clips in which our subject lemurs turned and looked at another lemur, they tended to orient their gaze subsequently, here in red, ahead of that lemur, as if they were following its gaze. And in fact, we can break that out over time. The gray pie is basically the distribution of gaze prior to, here in these time periods, prior to seeing the other lemur shift his gaze, and then the gray pie here is where our participant then looked after that. You can see that it's basically ahead of the other animal. Again, basically showing that in ongoing natural behavior between lemurs, that they also tend to co-orient in the same direction. That I think implies that this circuitry probably exists in much the same form in ringtail lemurs as it does in macaques and in people. So with the last couple of minutes here, I just want to turn to, this is not a lemur story, but it grows out of all this work that we did here in lemurs. You never know where science will take you. And at the time that we had just published these papers, I was approached by a graduate student in biology out at UC Davis who was fascinated by peacocks and their, the incredible display plumage that they use to court peahens. And it's been controversial since the time of Darwin, the purpose of the spots on the tails of these birds. And some studies suggesting that more spots yields higher fitness for males, and some studies saying that basically it has no impact. And this student said, well, maybe we should ask peahens what they are interested in. And we could do that using the technology that was developed here at the Lemur Center. And so we made some refinements to the camera system. Peahens are actually pretty large, so they can carry the backpack very well. And then here's our system here. We abandon the mirror and we use fiber optic cameras, in this case, to sample the visual scene and also to measure gaze. And we did this while peahens were visiting amongst males out at the Klopfer farm. We thank Peter and Martha for housing these animals out there, and they engaged in very natural behaviors. And what was really surprising, and this was a big shock to behavioral ecologists and evolutionary biologists, is that peahens don't give a whit about these eye spots, so they never look at them. They tend to just look along the bottom of the train of feathers, and which is really fascinating because it turns out that the width of that train is basically proportional to its length. And we know that males who have longer trains do better with females. And so in this case, size really does matter. And Jessica went on to show that, in fact, these eye spots probably serve a role in long-distance attraction. The females can see those eye spots over big hedges that tend to exist in the home environments of peafowl. And this, I think, raises some very interesting questions about the organization of visual orienting behavior in birds, of which we know basically nothing. Work from my colleague Eric Jarvis here at Duke over the last 15 years had revised kind of standard theories of the organization of the bird brain, which were initially thought that this big Wulst here was not like the cortex of primates and other mammals, but instead was like the striatum. And instead, now his revision suggests that it might in fact be the exact same thing. And so this raises the very interesting possibility that there may be a much wider vertebrate organization for social orienting behavior, acquiring visual information about others. And that's just a hypothesis because it has not been tested yet. So at this point, I've run out of time. I want to thank three people who were really instrumental in this. Bill Highlander for encouraging me to do this and supporting our efforts. Stephen Shepard, who did all of the orienting work, really developed this whole system for use in lemurs, and Jessica Yorzinski, who adapted it to peafowl. And of course, I need to thank the Lemur Center for all the help and all the work, and just it was a really wonderful place to work. And actually, it turned out that Bill and I split the cost of the eye tracker. So at that time, that cost about $25,000. And so I had a Sloan Foundation grant, and Bill split that with me. So technically, it still belongs to the Lemur Center, and I hope it's still here. And more recently, we've had some support from a Bass Connections project as well. And these are all of the various other organizations that have helped support the work over time. So thank you very much.